Hello and welcome back to the Farm Fundamentals, where we break down the complex drug mechanisms. Today we are starting a deep dive into autonomic nervous system. If drugs are the tools, ANS is the complex engine they work on. The ANS is the involuntary control center that keeps your heart beating, your lungs breathing, and your digestion moving without conscious effort. So it's a constant silent negotiation, and pharmacology allows us to hack into that negotiation. So in our segment 1, we deal into two opposing forces, that's sympathetic versus parasympathetic forces. So the ANS is primarily composed of two of these opposing branches, often called an antagonistic pair, that maintain a state of dynamic equilibrium in the body. So the sympathetic system is the gas pedal. So this is the fight or flight system. It prepares the body for action, stress, or emergency. It speeds up your heart, directs the blood flow to your skeletal muscles, dilates your pupils, shuts down the non-essential functions like digestion. It's designed to mobilize energy and resources immediately. On the other hand, the parasympathetic system, it's the brake pedal. So this is the rest and digest or feed and breathe system. It promotes maintenance, restores conservation, and relaxation. It slows your heart rate, constricts your pupil, stimulates salivation, and increases motility and secretion in your digestive tract. It's focused on restoring energy reserves. So together, they act like a nervous system version of thermostat, constantly adjusting the balance of bodily functions. And the segment 2, it's about the two neuron relay, or it's the functional anatomy. To control organs quickly and precisely, the ANS uses a two neuron relay to travel from central nervous system to the target organ. This relay involves a quick pit stop at a cluster of nerve cells called as ganglion. So the two neurons are preganglionic neuron. It travels from the spinal cord to the ganglion, and the postganglionic neuron. It travels from the ganglion to the target organ. That's maybe the heart muscle. So the chemical consistency over here is like in the both sympathetic and parasympathetic pathways, the relay begins with the same chemical signal. All preganglionic neurons releases acetylcholine at the ganglion. And the anatomical difference over here is that parasympathetic, so it has a long preganglionic fibers and short postganglionic fibers. The ganglia are typically located close or even inside the target organ. However, in the sympathetic system, it has short preganglionic fibers and longer postganglionic fibers. So the ganglia are usually located close to the spinal cord. And the chemical messengers, that is our neurotransmitters, we talk about them in our segment 3. While the preganglionic signal is always acetylcholine, the signal released at the target organ is what determines the system's action. So acetylcholine is the cholinergic signal. It acts, it's the primary transmitter for the entire parasympathetic system at the postganglionic level, that's at the end line. It mediates the rest and digest reactions. It's also used for all preganglionic neurons for both the systems. And norepinephrine, it's the adrenergic signal. It acts, it's the primary transmitter for the sympathetic system at the postganglionic level. It mediates flight or fright responses. So norepinephrine is primarily cleared from the synaptic cleft by reuptake into the nerve ending. And epinephrine, so it's the hormonal booster, or we call it adrenaline too. It's a hormone released by adrenal medulla into the bloodstream. So medulla is essentially a specialized sympathetic organ. It circulates widely, amplifying the effects of norepinephrine throughout the body to ensure a massive systemic stress response. So the receptor locks and keys, that's our segment 4. Drugs don't interact with the neurotransmitters, they interact with specific receptor proteins, that's locks, on the target cell surface. We group these into two major families based on which neurotransmitter they bind. So the cholinergic receptors or the ACH locks. So these receptors bind acetylcholine and are named after chemicals that originally helped identify them. We have nicotinic receptors located at the ganglia where the preganglionic fibers where the preganglionic acetylcholine acts, and at the neuromuscular junction in the skeletal muscle. So they are ion channels and mediate very fast signaling. Muscarinic receptors, so these are located on all parasympathetic target organs, heart, smooth muscles and glands. So they are G protein coupled receptors and mediate slower, more varied responses. So there are 5 subtypes, M1 to M5. Number 2, adrenergic receptors. So these the norepinephrine or epinephrine rocks. So these receptors bind norepinephrine and epinephrine. We have 2 types, alpha receptors and beta receptors. Alpha receptors, it's classified further into 2 types, alpha 1 and alpha 2. Alpha 1 primarily constricts the smooth muscles in blood vessels, leading to increased blood pressure. Alpha 2 often located presynaptically, acting as inhibitory brake on norepinephrine release, a feedback loop. Beta receptors, beta 1 primarily found in the heart, its activation increases the heart rate and force of the contraction. Beta 2 is primarily found in the lungs and skeletal muscle vessels. Activity causes bronchodilation and vasodilation. So to sum up, ANS is a dynamic balance between sympathetic and parasympathetic systems. All communications start with ACH, but the final actions are dictated by norepinephrine in case of sympathetic or acetylcholine in case of parasympathetic, binding to their specific locks, that's adrenergic or cholinergic receptors. Understanding these key receptors is the first step towards understanding how the drugs like beta blockers or muscle relaxants work. Next time, we'll dive deep into the specific effects of cholinergic drugs. Until then, keep it